Mitosis is a part of the cell where the nucleus divides into two identical nuclei, leading to the formation of two genetically identical daughter cells. In this video, the black sweatpants represent the cell wall, the silverware represents the nuclear membrane, the socks represent the chromosomes, the shoes represent the spindles, and the white headphones represent the spindle fibers. The cell starts out in interphase, which is where it spends most of its life. There are three stages in interphase, G1, S, and G2, which is when the cell prepares to undergo mitosis. G1 is the first gap or growth phase when the organelles are duplicated and the cell grows. In the next stage, synthesis, the DNA is replicated. In G2, the second gap, the cell makes proteins which are needed for the process of mitosis. When the cell is ready, it will go through mitosis, which has four stages: prophase, metaphase, anaphase, and telophase. During prophase, the nuclear membrane starts to break down. The sister chromatids condense, and the spindle, which helps organize and separate the chromatids, forms. Next, the cell goes through the metaphase stage of mitosis. This is where the sister chromatids line up at the equator of the cell. During anaphase, the spindle fibers pull the sister chromatid pairs apart. One set of the chromosomes is pulled to one side, while the identical set is pulled to the opposite side of the cell. Lastly, the cell goes through the telophase stage. The nuclear membrane starts to reform, and the chromosomes start to uncoil. After this, cytokinesis occurs, which literally means splitting of the cytoplasm, and the cell splits in two. These new cells will then enter the G1 stage of interphase, and the cell cycle starts over.